Hello, Professor.
Meet you.
Welcome.
Hi, hello everybody.
So Professor Andrea Fuso is an associate professor at Sapienza University of Rome, specializes in DNA methylation's role in late onset Alzheimer's disease. His work reveals how B vitamin deficiencies affect DNA methylation and gene expression, influencing neuroinflammation and amyloidogenesis. His research provides vital insights into Alzheimer's pathology and potential interventions. We greatly value your presence and eagerly anticipate your inspiring findings. So welcome.
Thank you, and I am really grateful for the opportunity to talk here, and congratulations for a very interesting conference. I hope you can see my screen.
Yes.
Okay. Just a moment. Okay, so yes, today I would like to show you how environmental factors are causally connected to Alzheimer's disease, to neurodegeneration. And to do that, I will focus on a particular epigenetic factor, which is DNA methylation. Sorry. Okay. So I will briefly introduce the epigenetics, some basics on the epigenetic mechanisms. Then I will show you how the environmental stimuli are connected to possible neurodegeneration through DNA methylation, so through epigenetics in the so-called environmental epigenetics. And then I will show you some results from my lab on Alzheimer's disease models as a paradigm for neurodegenerative diseases. What is epigenetics? I always like to start with this picture showing two different sculptures that are obtained from the same stone, from the same kind of stone, by an artist that used exactly the same instruments to form these two complex expressions. So epigenetics is actually manifesting different complex expression. We are talking obviously of DNA expression, mRNA expression, starting from the same raw material, starting from the same DNA. Epigenetics was defined for the first time many years ago in 1942 by Conrad Waddington. Waddington was studying the cell differentiation during embryonic development, and he defined the epigenetics as the branch of biology studying the causal interactions between genes and their products. And further, in the 90s, epigenetics was again defined as the study of the mechanism of temporal and spatial control of gene activity during the development. So as you can see, for many years, epigenetics was strictly linked to development, to cell differentiation. And still in the 90s, the definition by Holliday could include more or less any kind of intracellular connection, reaction, and interaction. Today, we have an agreement that limit the terms of the definition of epigenetic factors to few epigenetic traits. The most known are the DNA methylation, the histone modification. Together, these two factors are connected together with other protein factor to the chromatin remodeling, which is also considered as a third epigenetic factor responsible for the chromatin accessibility. And there are two other epigenetic traits that are not so universally accepted as epigenetics: the DNA hydroxymethylation, which is a further modification of the methylated DNA, but the transformation from the hydroxymethylation to a non-methylated DNA is very fast. So someone considered the hydroxymethylation as an intermediate of the demethylation processes, and the other studied factor really fashion in the last years are the non-coding RNAs, including in particular the microRNAs that can modulate again the expression of the messenger RNA. So as I told you, for many years and also today, the epigenetics was strictly linked to the differentiation of a cell, and this draft was made by Conrad Waddington in 1942. He imagined the differentiating cell like a marble rolling down a slope and taking different directions depending on the ridges and valleys during its movement. So the ridges and valleys represents the epigenetic factors that influence the cell differentiation. The marble usually stops when it reaches a point of low local elevation that represents in the Waddington view the differentiated state. Therefore, we can define the epigenetics like one of the driving forces in cell differentiation, responsible for the cell differentiation leading from one cell to a complex organism with different cell types, with different functions, with different phenotypes, and also responsible for phenotypic differences between individuals with very, very similar DNA. I would like to remember that more than 90%, almost 99% of our genomes is the same. And coming back to the cells, epigenetics is responsible for differentiation in many different tissues with different aspects and different functions. So why should we study epigenetics in a neurodegenerative processes? In a tissue like the brain, which has very limited cell renewal, which is a terminal differentiated tissue, well, we can still take advantage of the Waddington model and just imagine that when the marble are stopped on a platform along the slope of the mountain, then wind and snow and rain could still modify the platform where the marble are stopped and could induce a new movement and a new direction of the marble. This is the same for the cell. Even if terminally differentiated, the somatic cell can receive environmental stimuli that through the epigenetics can modulate its phenotype, which do not mean that the cell goes back to an indifferentiated state, but the cell could assume a new phenotype in terms of mRNA expression, so it can change its complex mRNA expression. Going in particular on DNA methylation, I would like just to remind you that DNA methylation is the binding of a methyl group CH3 on the carbon number 5 of a methyl cytosine, mainly in cytosine followed by guanine, CpG moieties, but not exclusively. And it is a physiological mechanism that it's used by the cells to regulate gene expression and to interact with the environment in a physiological manner. And we can, to be sure, we can define the DNA methylation as a way of transcriptional control. And just to establish an agreement between me and you, we have to take in mind that usually DNA methylation at promoter regions cause the reduced binding of transcription factors. Therefore, genes with methylated promoters are usually silenced or weakly expressed. On the other hand, the hypomethylated promoter that are fully accessible to transcription factor can be expressed in a discrete manner. So DNA methylation corresponds to gene silencing, hypomethylation to gene activation. Just one slide to point on a specific aspect of epigenetics. I would like to introduce this point, this concept, by explaining the difference between genetics and epigenetics. Genetic mutations cause the change of one base in the DNA, and it can be associated to loss or alteration of protein function if occurring in the coding region, or to alteration of gene expression is occurring in the promoter region, and it is irreversible. Epigenetic modifications are not a change of the sequence, but consists in the binding of chemical species to the sequence, like in the case of DNA methylation, that when occurring in the promoter region also cause the alteration of gene expression. But this mechanism is reversible. So we can imagine, I mean, the cell normally changes the methylation status to induce or repress gene expression. So we can also imagine to intervene by epigenetic drugs to modulate, for example, the methylation, and therefore to induce some expression change in the organism, in the cell. And there's a very great interest in this possibility, particularly in the field of the nutrition. And some time ago, I found this interesting device sold by a German company, proposed for the nutritionists that have to formulate a dietary protocol for their patients. And these instruments promise a very easy definition of the epigenetic traits. You can take an hair with the bulb from your patient, put it on this small device that check the electromagnetic waves generated by the hair, and in 15 minutes, a supercomputer in the company house can return to the professional, to the nutritionist, a panel of epigenetic modification that can be used to formulate the diet for the patient. That would be fantastic if it was the fraud. It is not so easy. Study the epigenetics, and I'm sorry guys, I'm a biochemist, and so I know these arts, but to study epigenetics we need to know something about cell metabolism. This picture represents more or less all the biochemical pathways in the cells, but I am a gentle biochemist and I would like to help. Talking about methylation, we are more or less here, and here means the so-called one-carbon metabolism. This biochemical pathway is actually more ramified. I've showed here just a small part of the one-carbon metabolism, which is the part related to DNA methylation, because the one-carbon metabolism, which consists in the transferring of monocarbonic units, is the pathway, the biochemical pathways that sustains the DNA methylation and other methylation. So if we consider the pathway starting from methionine, that is amino acid assumed by the diet, the methionine can be combined with ATP, forming SAM, the S-adenosine methionine, which is the main methyl donor in the transmethylation reactions. Transmethylation means that the methyl group is bound to other substrates, including DNA. After the transmethylation, SAM is transformed into SA, S-adenosyl homocysteine, which is an inhibitor of transmethylase reactions, but SA is rapidly transformed in homocysteine in the only reversible reaction of this pathway, and the reaction is not reverted in physiological condition, but is pushed through the let's say the clockwise reaction of the cycle, since homocysteine is rapidly discarded, or it is transformed through two mechanisms: the transsulfuration pathway on the left at the bottom, which uses vitamin B6 as cofactor and form a glutathione, which is the most powerful antioxidant in our body, and the remethylation pathway, which uses vitamin B12 and folate, another B vitamin, to remethylate homocysteine to methionine. So until this homocysteine is transformed or is scavenged, this pathway can function well and can methylate substrates. Let's go to the second part of the talk, and let's see how the environment can have its influence on this pathway and on DNA methylation and change DNA methylation. Talking of aging, we know that neurodegenerative diseases are the most risk for the aging, and actually we know that also for Alzheimer's disease, aging is the greatest risk factor for the onset and progression of Alzheimer's disease. We know today that the difference between normal and diseased aging can be modulated, influenced by environmental stimuli, and that environmental stimuli are mediated by epigenetics. So if we imagine that a post-developmental epigenome or methylome, which means the many DNA methylation in our cells is established at the end of the developmental period, then during all the life, and the longer the life, the longer the opportunity to encounter environmental factors, environmental stimuli, those environmental factors can hit our methylome and could cause a difference in the methylation at specific sites, causing a drift from normal aging to diseased aging. As you can see, many environmental stimuli can induce epigenetic modification, including physical stress and psychological stress, behavioral stress, of course, chemical species, and nutrition in particular, because it's the first way to introduce chemicals, compounds inside our body. When I was a student many years ago, few pathologies were related to epigenetics. Today, the number of pathologies that are associated to epigenetics is increased, and many of these are degenerative diseases, age-related diseases, and in particular, neurodegenerative diseases. Why moving from neurodegenerative disease to Alzheimer's disease? Well, the first unofficial reason is that I am a fan of this Italian cartoonist. The more official reason is that Alzheimer's disease can be assumed as paradigmatic for many neurodegenerative diseases. If we look at the aspect of the disease onset and progression, because it is a multifactorial disease, we are calling, of course, of the sporadic Alzheimer's disease, and it is the most present, most relevant neurodegenerative disease under the socio-economic aspects. When in my lab we started to study Alzheimer's disease, we decided to apply to formulate an hypothesis based on some data in literature, some clinical data showing that showing a connection, an association between high homocysteine, hyperhomocysteinemia, which means high homocysteine in the blood, low B vitamin levels, and low methylation potential. Methylation potential is the ratio between SAM and SA. As I've told you, if SA increases, methylation is impaired. So at the beginning of the 2000, the clinical association between these factors and the late onset Alzheimer's disease was evident, but the mechanisms were not. And there was a huge debate if, for example, hyperhomocysteinemia was a risk factor or a consequence of Alzheimer's disease. But since we are biochemists and we knew the one-carbon metabolism I showed you before, we hypothesized that high homocysteine and low B vitamins could be connected. If we have low B vitamins, the transsulfuration and the transmethylation pathways are impaired, so we have a rise in homocysteine, so we have a rise in SAH, and so we can have low methylation potential, a general methylation impairment, and methylation impairment could cause altered gene expression. So we decided to explore the possibility that genes involved in Alzheimer's disease could be modified by high homocysteine and low B vitamin. The idea that Alzheimer's disease, and again, we are talking of late onset Alzheimer's disease, the slow and late onset form of the disease, could be caused, or at least could have as a cofactor epigenetics mechanisms, was clear by the observation that many risk factors, and this is just a short list of the risk factors involved in Alzheimer's disease, could be related to environmental stimuli, and this non-Mendelian etiology strongly suggests that epigenetics could be at least involved in the onset and in the progression of the disease. And as a matter of fact, in the first decade of the years 2000, we observed a number of reports showing that cognitive disorders are associated with epigenetic dysregulation, showing that DNA methylation in individuals, in the brain of individuals, changes over time, generally toward hypomethylation, that DNA methylation was altered in neurodegenerative diseases and could be area specifics. So we have a number of observational data showing that there's an association between epigenetics, methylation in particular, and Alzheimer's. Just to show you two examples, this is a study that evidenced the drift in the DNA methylation of different genes involved in Alzheimer's disease. I would like to highlight the last gene on the right, PSEN1, which is hypomethylated in Alzheimer's disease. And another interesting observation was made on twins, identical twins. So these individuals had completely identical DNA sequences, but one of the two developed Alzheimer's disease in association with alteration of DNA methylation because of environmental exposure. In particular, the twins reported in this study, one of the twins reported in the study, the one with Alzheimer's disease, worked with pesticides. So we have to go for a moment to see what are the main molecular mechanisms known in Alzheimer's disease. Of course, the amyloid processing, which is the proteolytic cleavage of the amyloid precursor protein by gamma and beta secretases, leading to the toxic amyloid beta peptide that accumulates in the cell plaques. The beta secretase is encoded by the base1 gene. The gamma secretase is a multi-protein proteolytic complex in which the active site is encoded by the PSEN1 gene, the one I showed you before. As for tau phosphorylation, we know that besides amyloid plaques, the Alzheimer's brain also show accumulation, intracellular accumulation of hyperphosphorylated tau. Tau is a microtubule-associated protein that can form neurofibrillary tangles when hyperphosphorylated, altering the microtubule equilibrium. Then we know that inflammation is involved in Alzheimer's disease. In a double way, inflammation is triggered by the presence of amyloid species, but also the neuroinflammation deriving from other situations, for example, stress or chronic stress, or for example, trauma, can induce the aggregation of amyloid beta. And the same for the oxidative stress, because the oxidative stress is triggered by amyloid beta deposition, but can also induce tau hyperphosphorylation and amyloid processing. So those are the main molecular pathways, molecular mechanisms that we should observe when studying Alzheimer's disease. And today it is clear that there's not a single cause of the Alzheimer's disease onset. For many years, the researchers focused on amyloid or on tau. Now we know that we have to look at the whole picture and to take into account all these molecular mechanisms when we try to find out some molecule that could help in the treatment of Alzheimer's disease. Therefore, going back to the, sorry, just one moment. Going back to our model, this is the same pathway we showed you before, the one-carbon metabolism, just a little bit revisited, and we decided to modify the one-carbon metabolism by a nutritional intervention, which means that we decide to give our transgenic AD mice, the TG78 are a model of Alzheimer's disease, a genetic model of Alzheimer's disease. We decide to use these mice for their rapid amyloid beta plaques development and to raise the mice with a diet deficient in folate, B12, and B6, therefore reducing the capacity to transform homocysteine in the transsulfuration in the remethylation pathway, with the aim to induce hyperhomocysteinemia and methylation potential impairment. On the other hand, we supplemented the mice directly with the methyl donor, SAMe, exogenous SAM, with the aim to revert the effect induced by B deficiency. We also used the opportunity to study some of the molecular pathway in post-mortem and human blood samples, post-mortem brain tissue and human blood samples, but of course we could not intervene on the one-carbon metabolism in these subjects. There are not yet clinical trials active with B deficiency or with SAM in Alzheimer's disease patients. Therefore, these reports show the results in the mice brain, in the TG78 mice brain, with B deficiency and some supplementation. And as you can see, homocysteine increases with B deficiency, confirming our hypothesis, and is restored. It converts to control-like levels with SAM. And on the other hand, the methylation potential, the SAM-SA ratio, is reduced by B deficiency and improved by SAM supplementation. In order to study the effect of this alteration on DNA methylation, we had to study the methylase and demethylase activities. So we know that in our cells we have a maintenance DNA methylation activity, which is exerted by the DNA methyltransferase 1 that can reproduce DNA methylation during cell reproduction, cell replication, sorry. When DNA methyltransferase 1 is inhibited, we have, or the methyl groups are poor, we have a passive DNA methylation during cell replication, but we also have a de novo methylation activity exerted by DNMT3A and 3B, and this can be particularly relevant in the brain where there's few cell replication. And an active demethylation pathway, which is mediated by the TEAD proteins that pass through hydroxymethylation. And studying the enzymatic activities in the brain of our mice, we could confirm that the methylation impairment caused an impairment in the maintenance and de novo methylation and an increase in the demethylase activity. And all these mechanisms were corrected by some supplementation. The third step to connect the nutritional intervention, the environmental intervention with the disease was to prove that the alteration of methylation could hit some specific genes involved in Alzheimer's disease. We studied many of the genes involved in amyloidosis, in tau phosphorylation, and so on, and we found that one of these genes was modulated. How to study DNA methylation? The gold standard today is the transformation of the genomic DNA by bisulfite that transform cytosines in uracil unless the cytosine is methylated. Methylation protects the cytosine from this chemical reaction. So at the end of the reaction, we have a transformation of the non-methylated cytosine in uracil. If we amplify the sequence of our interest, we will have at the end a C to T substitution in our sample, and we can sequence our samples and individuate the specific site in which we have methylation or demethylation. And I would like to stress that this technique allows us to study every single cytosine in a determined region. We choose the promoter of a gene, we amplify that promoter, that specific sequence, and we can profile exactly the methylation status of each cytosine in this gene. Applying this technique, we found that presenilin-1 gene was modulated by methylation. As you can see here, 4 proximal CPG sites. Proximal means that they are close to the transcription site where the mRNA starts to be formed, were hypomethylated by B deficiency, and some could revert the hypomethylation. The changes in methylation, as expected, were associated with the changes in mRNA expression. If you remember, I told you that hypomethylation B deficiency is associated with increased expression, whereas methylation in some supplemented condition is associated with repression. So we could observe SEN1 overexpression by B deficiency and repression by some supplementation, both at mRNA and protein level. And this, as a consequence, a change in the amyloid plaque deposition. In the upper picture, you can see the normal condition of these mice, of TG78 mice at 3 months of age. In brown, you have the amyloid plaques. You can see that the plaques are increased with the B deficiency and reduced by some. And we also studied the behavior of these mice by the water maze test. And in particular, I think you are already all more expert than me in the behavioral task and behavioral assessment, in particular in the QA task, which is in some way an indication of the short-term memory. The B deficiency and the some supplementation could significantly alter the capacity of the mice, the memory of the mice. Therefore, in the end, we had the opportunity to link a nutritional and environmental intervention, B deficiency or some supplementation, with the symptoms, with the Alzheimer's-like symptoms in the mice through specific and well-characterized molecular mechanisms. What about the other factors? I've told you that amyloid production cannot be considered as the only responsible for Alzheimer's disease onset and progression. And I must say that we did not find any alteration in other genes. We found an alteration in the expression of BACE1, which is the beta secretase, so the other enzymes responsible for amyloid processing, but BACE1 is not directly, it is regulated by the alteration of the work-number metabolism, but not directly by DNA methylation at its promoter sites. And as for tau, we cannot find any gene alteration induced by methylation, but one of the proteins involved in tau phosphorylation is modulated by methylation at protein level. In particular, the PP2A protein, which is a phosphatase, which is able to remove phosphate groups from tau, and so to hamper the hyperphosphorylation, to prevent the hyperphosphorylation, the tau aggregation. Well, the PP2A protein, the activity, of the PP2A protein is increased by protein methylation. And in our mice, we could observe that B deficiency was responsible for decreased PP2A activity, and some could restore the correct activity. And this was linked to increased tau phosphorylation in B deficiency and decreased tau phosphorylation in some supplementation. These mice do not accumulate neurofibrillary tangles, so we could not measure the spreading of the neurofibrillary tangles, but we could check the phosphorylation at the tau protein. Therefore, at the end of this first part of our studies, we were able to define that the nutritional intervention, B vitamin deficiency, could impair the transphosphorylation and the remethylation pathways, causing hyperhomocysteinemia causing increase of SAH that is an inhibitor of the DNA methyltransferase activity and a promoter of the DNA demethylase activity. This alteration in particular could lead to the altered methylation in SEN1 promoter, leading to increased gamma secretase activity and increased amyloid deposition. Of course, as we have seen, some supplementation can contrast the B deficiency effect, and therefore we are aimed to at studying the effect of some supplementation in humans, and the first trial with some is started 1 month ago in Australia, and we are eager to know about the effects of some supplementation in mild cognitive impairment subjects. One of the questions I expect when I present this data is, okay, you give some to the mice, you found the alteration of one or few specific genes, but what happens to the whole genome? We could expect that altering DNA methylation could have an effect on many genes. To check this aspect, we decided to study the transcriptome and the proteome of the brain in our mice, and surprisingly, we found that, for example, in this microarray assay to study the expression of 600 genes of the normal CNS metabolism, only 1% of the genes, which means 6 genes, were modulated by B deficiency and some. And in this whole proteome on the right, we found only 16 proteins modulated by B deficiency and some. Why this is possible? Well, our idea is that this kind of intervention on DNA methylation passes through a physiological pathway. We are not supplementing with drugs that ex abrupto stop, impairs, avoid DNA methylation. So with a mild modification of the pathway of the 1-carbon metabolism, we probably act only on that genes that are normally modulated by DNA methylation in the adult brain. This is our hypothesis. But we also know that Alzheimer's disease is manifest during the adult age, but probably as more ancient onset in young individuals. And so we try to see what happens if we supplement mice during the embryo life, because we know that the early life is the most sensitive window to the environmental factors. So we performed these experiments in which only the mothers were fed with some supplementation. So the pups were supplemented during the perinatal period, from the mating during the pregnancy until the weaning, and we could compare the effects of the post-weaning some supplementation, which is what I showed you before, with the perinatal supplementation. And as you can see, the two highlighted boxes represent the all-C methylation in the promoter of SEN1 in mice. The B panel represents the methylation profile in mice fed with SAM after the weaning until the adult age. In the D panel, you can see the methylation profile of the mice that only received SAM through her mother, and you can see then they have not did not have some supplementation during the rest of the life. As you can see, the methylation profile is more or less the same. The methylation established by SAM is maintained during the adult age, and as you can see in the mice fed with SAM during the perinatal period, the two panels below, we have the same reduction in amyloid deposition as in the mice supplemented post-weaning. And you can see here, following the green and the red light representing the CpG and total methylation, that the methylation which is established at the weaning is then more or less maintained until the adult age. And then also this short term, some supplementation during the perinatal period is at least as effective as the longer and continuous supplementation after weaning. And this allows us to introduce the concept of epigenetic cellular memory, in which the epigenetic changes are induced in the embryo and then are maintained until the adult age. So this stresses the possibility to check and to, in case, supplement the individuals with methylating agents to prevent or to delay the onset of the pathology. As I told you, we had the opportunity to check the DNA methylation in the brain of normal and Alzheimer's disease, normal individuals and Alzheimer's disease patients, and in collaboration with two groups at the University of Barcelona and University of Amsterdam, in a work that in which also Maria Teresa Fiorenza was involved, and we collaborated in this part of the story. We could observe that in individuals, and I would like to remember that here we don't have neither BEAT deficiency or supplementation, but in normal individuals, if we check the SEN1 methylation in the brain of control subjects, we can see that Alzheimer's patients in the early phase and in the late phase of the pathology have less DNA methylation, in particular less methylation at the non-CpG sites. And it is interesting to point out that the methylation at the non-CpG sites is the one exerted by the de novo methylphosphatases, DNA MT3A and 3B. And also the expression of SEN1 in these subjects was in agreement with the methylation. As you can see in the Alzheimer's disease, we have an increased expression of SEN1. In this project, we also had the opportunity to check some specific aspect on neuroinflammation, and now we are following the studies on pro-inflammatory cytokines in the preclinical model. But in this case, we started from the clinical model. We used the brain of the subjects, Alzheimer's conscious subject I've showed you before, to assess the expression of a plethora of pro-inflammatory cytokines. And we were particularly interested in two molecules, EL1 beta and EL6, that have a particular profile. For example, here EL1 beta is highly expressed in the first phases of Alzheimer's disease in light gray, and then the expression of EL1 beta decreases with the progress of the pathology. The same profile can be observed for EL6, also if a lower extent. And so we decided to check the methylation of these two genes, and we could observe, in fact, that DNA methylation of their promoter and mRNA expression are very well correlated, because we have a huge hypomethylation in the first phases of the pathology, where the expression is higher, and a restoring of DNA methylation in the last phase of the pathology, when the expression is reduced. The same, more or less the same profile for EL6. In this case, we could not observe a restoring of DNA methylation, but the EL6 methylation continued to decrease during the stages of the pathology. Again, here we don't have any epigenetic intervention. Then in this case, we moved from the bedside to the bench side to check how the alteration of the one-carbon metabolism could potentially modify the expression of these genes. So it is clear now that SEN1 is not the only responsible, it's not the only gene altered in association with Alzheimer's disease. I've already showed you EL1 beta and EL6 associated to neuroinflammation. But as I told you, also if we just look at the amyloid processing, we need two proteolytic activities, gamma secretase and beta secretase. Beta secretase, I told you, is encoded by base1, and base1 have the same modulation as SEN1 in our mice. It is induced by bit deficiency and reduced by SAM, but we could not find any difference in the methylation of this promoter. So very recently, this work is still in preparation, will be submitted in the coming months, I hope. We decided to explore the effects of the alteration of one-carbon metabolism on another epigenetic mechanism, which is the the microRNA expression. So we assessed the expression of many microRNAs under some supplementation and bit deficiency, and we could, in particular, individuate one specific microRNA, miR29A, which is associated to Alzheimer's disease. It is considered like a protective factor in Alzheimer's disease, and this microRNA is regulated in the opposite way respect to base1. It is induced by SAM. It is unexpectedly induced by methylation and reduced by B vitamin deficiency. And what's interesting is that one of the targets of this microRNA is exactly base1. So you can see that when the microRNA is overexpressed with SAM, base1 is repressed because probably there's there's more binding of this microRNA to the base1 mRNA. We confirmed this mechanism by loss of gain of function experiments with microRNA transfection in cell cultures, and we also assessed the methylation, in this case not of the promoter, because the microRNAs are not coding genes. In this case, the miR29A locus is on the minus strand of a different gene. So we checked directly the methylation of the region coding for the microRNA, and we found that the methylation in this, as expected, observing the expression, we observed that the methylation is still as we expect. When we supplement SAM, we have a rise in the methylation. When we deplete B vitamins, we have a decrease in methylation. But in this case, we are looking at the methylation in the coding in the region that is not the promoter, but the region which directly allow the synthesis of the microRNA. And we know that when the methylation occurs in the gene body instead of in the promoter, this can be associated to a more stable expression of that specific sequence. So what's interesting here is that DNA methylation and microRNA expression are connected, and there's probably a crosstalk between DNA methylation and microRNA expression. This data, the data on the DNA methylation and the data on microRNA also give us the hypothesis to further develop the studies to explore if these factors can be used as a blood biomarker. In particular, for DNA methylation, we could individuate a specific stretch of CCTCC, which seems driving the changes in methylation both in SEN1 and in EL1 beta promoter. And for the microRNA, we also developed in collaboration with a group in the University of Naples a device that could, like the test for the COVID, a paper strip that could allow the detection of the circulating microRNA. So we would like to propose these two epigenetic factors as biomarkers, secret biomarkers of Alzheimer's disease. Again, to go to the last of the molecular mechanisms involving Alzheimer's disease, the oxidative stress, This is a story that just started. We are now starting some experiment directly with glutathione, but I would like to remind you that the human carbon metabolism is correlated to the production of glutathione, which is the most potent antioxidant in the cells. And in mice, we could observe that some supplementation could could cause an increase in glutathione production, whereas B deficiency reduces the glutathione production. So in the end, we can depict a more complex idea of how DNA methylation, and in particular, some supplementation is involved in Alzheimer's disease. We have already seen how some supplementation can reduce SEN1 expression via methylation and so reduce senile plaques. We now discover that DNA methylation could induce the expression of the miR29A that inhibits base 1, so reinforcing the SEN1 action and so contributing to reduce the amyloid plaques deposition. We also know that methylation of protein level can increase the PP2A activity, so decreasing tau phosphorylation, acting therefore on neurofibrillary tangle deposition, and that the effect on EL1 beta, EL6, and probably other pro-inflammatory cytokines, reducing the expression of these cytokines, can in turn can contribute to the control to the reduction of the senile plaques and neurofibrillary tangle deposition, as well as the increasing of glutathione synthesis. Therefore, we are depicting a central role for DNA methylation in controlling different aspects linked to the Alzheimer's disease onset and progression on the molecular point of view. I finished my talk. I would like to again thank you for the invitation. I would like to mention my group and the many collaborations we had in this year to perform this work, both in Sapienza University and with other Italian and foreign universities. If someone of you should be particularly interested in methylation after this talk, I would like to invite you to check the site of the Epigenetics Society. It is a new site, the Epigenetics Society. We organized for the first time the International Conference of the Epigenetics Society 2 weeks ago here in Rome, and on the website you can find many information on epigenetics. The association is free for young researchers and will allow the sending of a monthly newsletter with nice information on epigenetics. And I found my presentation, and thank you for the attention. Questions are welcome, of course.
Thank you, Professor, for this instructive presentation. We actually got some few questions. So the first one is from Catalina. She says, thank you, Professor Fuso, for your presentation. My question is, are there specific lifestyle factors or environmental exposures that can promote or mitigate DNA methylation changes associated with late onset Alzheimer's disease? And how might this knowledge inform preventive strategies?
Okay, if we intend lifestyle in general, yes. Well, some years ago, I was at the Alzheimer's disease conference, and the keynote speakers had a talk on the preventing Alzheimer's disease. And the prevention was like my grandma's suggestion. So eat well, do physical activity, maintains your brain active, and it seems silly, but it works. And actually, today we are observing a slight modulation on the onset, on the prevalence curve of an Alzheimer's disease, which is considered to be associated to the many preventive actions that are suggested, for example, for cardiovascular disease, but are in common with Alzheimer's disease. So for sure, all these good granular suggestions are useful for to attempt the prevention of Alzheimer's disease. We are not yet on the possibility to say takes this in supplement and you will delay or you will not have Alzheimer's disease. Let's see what's happened after the conclusion of this first trial with Sam. But at present is still a possibility that supplementation with the epigenetic agents can help.
Okay, thank you. We've got also another follow-up question. So epigenetic changes may originate early in life, but become manifest in old age. What is the significance of this phenomenon in the context of neurodegenerative diseases, and how might it impact our understanding of disease prevention?
Yes, this is absolutely correct, and this is the reason why we decided to explore the possibility that early life intervention could be effective also in late life. This is, of course, something we can control less, but the developing period, the embryo life, is the more subject to epigenetic changes, and therefore we, of course, can hypothesize that alteration in the epigenome during the early life can be associated to neurodegenerative disease. There are many studies showing, for example, that alteration in the nutritional lifestyle, in nutritional style, and in particularly when we have a difference between the in utero nutrition and the postpartum nutrition, when, for example, individuals are in the sort of restricted caloric restriction during the pregnancy, during the in utero nutrition, and then they move to a fat-rich nutrition. Here we have the highest drift and the highest possibility that the epigenetic changes can induce a general, not just for Alzheimer's, but a general alteration in leading to disease. Yeah.
Okay, thank you very much, Professor. Thank you for your presence with us.
Welcome.
Thank you. So now we're gonna proceed for our last talk before lunch break. Introducing Professor Claudio Babiloni.